Welcome to Didactic. I'm your host, Dr. O'Leary, and today I'm going to be talking about Bayesian networks, especially in our brain, and kind of demonstrate how our brain is a Bayesian calculator, whether we want it to be or not. So here are my disclosures. You can pause and read them if you want. Today I want to explain why Bayesian thinking, though it's really historically been neglected in lots of different kinds of sciences, but especially in psychiatry, is super important for understanding how we implicitly have biases that we don't even know about. Also, I want to explain how the basic concept of small world, scale-free attractor networks applies to potential psychopathologies in our brain, or more generally to just how our brain works. But I want to start with just evaluating knowledge in general. I mean, how do we know what we know? One of the first things we think about is plausibility, and that's basically that whatever we're proposing does not obviously violate logic or seem to have a very low probability of being correct. Also, we think about plausibility in terms of mathematics and science. If something can be modeled mathematically, then it seems to have a priori a more reasonable chance of being true. It's also generally more testable if it can be modeled mathematically. And if there are proposed mechanisms that, say, violate established theories that have a lot of support for them, then those would really require some extraordinary evidence to be taken seriously. They would have very low prior probabilities of being true. Another concept that's been increasingly important is consilience. So the more knowledge that we have, the more we're able to confirm that different types of knowledge agree with each other. For example, in the sciences, the mathematics, or the humanities, then anything that we have that kind of disagrees with any of those things is not consilient. And so we would think that it has less chance of being true. For example, there are energy therapies that break the laws of physics. This is an example of Reiki. It's an AI-generated image, so these are not real human beings. And there are magnets. Both of them claim to be manipulating energy fields around human beings. But you might think, well, magnets are far more likely to have an actual effect on, say, pain than channeling cosmic energy or the God consciousness. So you've just proposed a prior probability. A lot of what we do, especially when we do clinical trials, results in just getting halfway there. We reason through that something is likely to be true based on clinical trial results, but we only do it halfway. Clinical trials do not produce probabilities that the results are real. Instead, what they produce are estimates of magnitude of effect of something, a treatment, and whether or not the result is likely to be from a random sample of some assumed distribution. It's not a probability. Now, failing to calculate posterior probabilities that the results are true is basically assuming that everything we're testing is equally likely to be true a priori, and that's a problem. So imagine yourself saying, the prior probability that clinical trials confirming the efficacy of therapies that break the laws of physics is equal to the prior probability of therapies that do not break the laws of physics. But that's what we're doing when we simply compare them without including Bayesian math. So Bayesian reasoning is basically a way to test for confidence that the magnitude of effect is real. Not just a p-value, but actual confidence that we can have that the result is real. So the results have to be converted into likelihood ratios, and then those have to be multiplied by the prior odds that something is true. A prior is just an estimate of bias, whether that bias is right or wrong. So in clinical tests, we estimate bias by the population prevalence of whatever it is we're testing for. Say that's COVID-19. If the prevalence is very low in the population, then we would expect more false positives on the test, for example. Other factors might be considered too, like the effectiveness of blinding, the risk of publishing bias, the risk of industry bias, the plausibility of the mechanisms, which I just discussed earlier. All of that can be taken into account to estimate a prior probability. So you do all this and you do the math. You end up with a posterior odds, which can be converted into a posterior probability that the results are, in fact, true. We do this because there is always a risk of getting false positives. Think about the Reiki versus the magnets. If we were to conceptualize this as, if the null hypothesis is true, then Reiki is not effective. If the alternative hypothesis is true, then Reiki is. So you can estimate a prior odds that the null hypothesis is true versus the alternative hypothesis. And in this case, you can do like 999 to 1. And this is really a very generous estimate, given that the actual odds of something that breaks the laws of physics are about infinity to 1 or approaching 0 probability. But we're going to be generous because we need to be able to do some math. So we're saying basically, like, if you do 1,000 trials, we expect 999 of them to give false positive results. For the magnet, you might say, well, I don't know that magnets really have that much effect on pain, especially if they're just passed over the body, but they are plausible, so I'm only going to give them a 2-to-1 ratio. Now you have to do a little bit of math. We start with the alpha, which is the false positive rate at which we are comfortable when we do the test. There's also beta, and that's the false negative rate. So does your test have enough power to find a difference if that difference is small? And then we have pi here, which is the prior probability that the null hypothesis is true. So basically, if we set our alpha at 0.05 and our beta at 0.01, our false positive risk is 555 to 11, or you could also think of that as it's basically 50 times higher than just 50-50, or 1-to-1, which is what we assume test results mean if we're not adding priors. So the probability in this case, using these numbers, that you're getting a false positive is 0.981, or 98.1%. So even though we know that we would get mostly false positives, or really all false positives, if we were to do that study, some people do the study anyway, and then they get positive results. So then they say, well, look, Reiki's determined to be as effective as a well-researched, say, chemotherapy, for example. How confident can I be that that is a true positive? And I included this image here because I asked an AI to generate an image, and I gave it the prompt, a face palm, and this is what it gave me, a face and a palm missing a finger. But getting back to Bayesian reasoning, if I were to take this equation up here, which is the basic Bayesian equation, and just say it in words, it's the odds that A occurring, assuming that B is true, equals the odds of B occurring, assuming A is true, times the odds that A is true divided by the odds that B is true. But we can simplify this down. So this part of the equation here, the probability that B is true, assuming A is true, divided by the probability that B is true, gives us something called a likelihood ratio. And a likelihood ratio is an odds ratio. So if you simplify it down in language that I can understand, basically your posterior odds equals your likelihood ratio times your prior odds. So you can look up how to do this from data that's already been reported in clinical trials, but they didn't report likelihood ratios, and that's in the Perneger 2021 paper here at the bottom if you want to look it up. So you take your prior from prior knowledge, and we use the odds to do all the calculations. So for example, if your prior was 1 to 999, and that's that you would get 1 true positive for every 999 false positives, then your odds is 1 out of 999, and that converts to 0.001001. If your likelihood ratio ends up being 5 in your study, and you plug that number in to this equation up here, then you get a posterior odds of 0.005005. So very, very low odds that the result is actually true. That's about 0.49%, or about half of 1%. Now, if you had a very high likelihood ratio at the end of the study, that meant that the data It's very supportive of the magnitude of the effect that you estimated. So you could get a posterior odds, in this case, of 1.001001. So that's basically just 1, which is a posterior probability of about 50% that your results are actually true. So even with this super high likelihood ratio, you still have only like a 50-50 chance that the results of your study are actually true if you assume these numbers. Now our brains are Bayesian calculators. They have built-in priors. These priors can be referred to as like expectations or our default settings or predictions. Now these are based on evolution, like our genetics. They are just written into us, but they also can be made because of our experience, environmental factors. And then every once in a while, random processes come in and just mess everything up. So now I'm going to get to our network models and try to tie all this together. So now I'm going to start with some very simple neural networks. Cortical columns are organized into layers, and they form networks that process afferent and efferent signals. They call these layers in the brain basically columns when they have neurons that are working together. And most of the cells there, about 80%, are excitatory cells or glutamatergic pyramidal neurons. Now, among these pyramidal neurons, there are inhibitory GABAergic interneurons, and they act to modify the local network. They can modify directly the pyramidal cells by reducing their rate of firing, or they can modify each other. So you can inhibit an inhibitor, and that results in more firing in the pyramidal neuron because it has less inhibition. Now, pyramidal neurons are exciting each other in different layers. So they're talking to each other basically in this column, but we're also getting signals from outside of this column. So sensory information or information from other cortical regions, and especially information from like thalamic regions are coming in to modify the activity of this cortical columns are organized into layers, and they form networks that process afferent and efferent signals. They call these layers in the brain basically columns when they have neurons that are working together. And most of the cells there, about 80%, are excitatory cells or glutamatergic pyramidal neurons. Now, among these pyramidal neurons, there are inhibitory GABAergic interneurons, and they act to modify the local network. They can modify directly the pyramidal cells by reducing their rate of firing, or they can modify each other. So you can inhibit an inhibitor, and that results in more firing in the pyramidal neuron because it has less inhibition. Now, pyramidal neurons are exciting each other in different layers, so they're talking to each other basically in this column, but we're also getting signals from outside of this column. So sensory information or information from other cortical regions, and especially information from like thalamic regions are coming in to modify the activity of these cortical neurons. We also get modulatory activity from the brainstem, including the ventral tegmental area, and this dumps dopamine into the system, and that can affect the rate of firing of the different neurons. There's also serotonin from the raphe nuclei that get dumped into the system, and a bunch more neuromodulators either produced locally or at a distance like norepinephrine that can modulate how this network works. All this is to say, really, I can't handle the truth. I mean, even in a vastly simplified model, my brain is not capable of understanding the extents of all these interactions, but that is okay. That's where network models come in. So I'm going to start by talking about small world networks. Small world networks, if you think about people, are basically networks where you're connected to everyone else in a very small number of steps. So you have all of these nodes, and nodes could be humans, and you can get to any other node in just a few steps. They say that all humans have like 6 degrees of separation. If you were to randomly pick someone and they ended up coming out of Saudi Arabia, you'd randomly pick somebody else and they're in the Philippines. Well, you can connect them somehow in 6 or fewer steps, basically. And that makes your network a small world network. A node is just an integrated unit. I think in mathematical terms, you would say that it has a Markov blanket around it. There's an internal state of that node, and then there's external stuff. And this can be at different levels. So nodes can form communities or cliques, which are groups of strongly interacting nodes. And those communities or cliques at some level could be modeled as nodes themselves. But then you have hubs in a network, and a hub is a node that is really highly connected to all the other networks. So you have some of your connections, in fact, most of them have very few connections to other things. So small world networks can be called scale-free networks. And that's because as the fraction of nodes increases, the number of connections that you expect from that fraction is going to go down. What this figure basically means is very few nodes have a lot of connections, and a lot of nodes have few connections, and then there are just some in between. The nodes that have lots of connections we call hubs. Now we have hubs in the brain. So this is from Oldham et al. In 2019, and they basically identified areas in the striatum, in the thalamus, in our central executive network, our default mode network, our memory networks, our salience network, and visual networks that form major hubs in the brain. And that means that these areas are more connected by whatever cutoff they used than most of the other areas. So that kind of begs the question, how are these networks formed? I mean, we expect genetics to play a role, right? There's this broad network design that's designed into our genes, and our networks have to function in a way that helps us to survive immediately when we're born. But we also develop. So we have experiences, the environment affects us, and there are just chance things that happen while we're developing. Neurons that fire together wire together. So basically, as you are experiencing the world, some neurons are going to fire together more than others based on your sensory inputs and your feedback from your behaviors. And so these neurons are going to wire more strongly together, and that is going to maintain and isolate certain networks from others. Now think about how long it takes an infant just to develop the basic motor skills to, say, pick up a Cheerio and put it in their mouth. Like, this is very hard. Part of that, they're slow because there's a myelination process going on, but also just the coordination and function of making a behavior in their brain is developing, and it takes a long time for them to, say, move their right hand without moving their left hand or their foot at the same time. Now, during adolescence, after we've built up a whole bunch of networks and they've somewhat solidified but are still kind of leaky, there are these massive pruning processes where neurons have a difficult time making new connections, but a lot of connections are lost. So we think this helps to just solidify these networks. We end up with beliefs and personality features that are really hard to change. It's difficult to change someone's beliefs. It's difficult to change their personality. They can. There is some flexibility, but it's not nearly the kind of flexibility that children have. Another thing that is super important about networks is that they take complex information and they integrate it into one or a few signals. So what this means is that in a network, decisions are basically made by committee. And every once in a while, some node goes rogue and sends false signals. But in general, decisions take complex information and they send out relatively simple information. So imagine you have this big network here at this level, and it is part of the same kind of networks in different parts of the brain. Those integrate all the information and they send it to higher level networks. Those higher level networks work together to create things like behaviors in humans. Human behaviors can be integrated together in our groups and in our institutions, and our institutions can be said to have behaviors. But all the information that started up here just to make these neurons do something has been lost by the time we get, well, to this level, really. And this level seems almost not connected to this level at all, but it definitely is. So as you move from one local network node to another, or one scale to another, the local information that complex information, it's lost because the input is integrated into simpler output. So you have these inputs, and just imagine trying to move your leg. I mean, it requires thousands or millions of neurons to initiate the process. But when you move your leg, there's just a few neurons involved in flexing those muscles. So that information gets sent to another network, but it gets sent out having lost a lot of the information that came in. There's also the possibility of gaining information. So basically you're not getting an input signal, but it's still sending an output signal for some reason. So we could call this misinformation. And this is what we think results in things like illusions or hallucinations or delusions. So we can conceptualize our mental life at different scales. Our medical model basically looks at things at the biochemical and the cellular, tissue, organ, and sometimes whole organism type level. Our psychological and psychoanalytical models look at whole brain level and whole human level behaviors. And then our more sociologically based or ecological models, they look at the organization of multiple behaviors among humans into the behavior of, say, institutions and how these affect individuals. Now I'm going to talk about how these small world networks can be organized into attractor networks. So attractor networks are basically networks that create stable states. According to Looijestijn et al., I'm not sure how you say that, but you can find the references in the notes. They state that attractor networks are characterized by a more or less pronounced attractor state, which is a preferential low energy state toward which the network tends to converge by default. So these low energy states are characterized by kind of just noise. You have a low frequency firing patterns and some excitability, but the downstream networks are really unlikely to respond to this random firing. They know that this firing isn't actually sending any real information. I think of a metaphor as like kids are quietly watching a movie in the other room, and every once in a while there's an occasional squeal. Mom thinks about it, but she's like, everything's fine. I don't need to interpret that as anything important. But then there are high energy states, and high energy states happen when enough energy is suddenly added to the network. So you could say glutamate release in neuronal terms, and it becomes highly active. And it has robust excitatory signaling and inhibitory signaling if that's what the network does. So then you think about, well, the kids were quietly watching a movie and then suddenly pizza arrives and the state goes crazy for a while. But after they eat, they basically return back to this low energy state. So we think of these states as basins. So an attractor basin you can think of as the low energy basin here where the green crescent is. But if you put enough energy into it, say activation energy, then you can get it into a high energy basin. To get out of the high energy basin, enough energy has to be present to push it back over that hill. And that energy is usually available just by random noise. And that can push it back down into the low energy basin once it's done sending its signals. So you can excite, get up here, you can de-excite basically, or inhibit and go back down to your low energy state. Now the energies required to change these states act like built-in Bayesian priors. So why does that matter? So basically priors determine the rates of false positive and false negative signals. So your network is always sending a little bit of signal, but it's not being interpreted as a real signal. It's not causing other networks to go to their excited state. If you have a very high energy of activation, you're going to have very few false positives because no information is going to get through unless it has a lot of force behind it. But you'll also get a lot of false negatives. You won't be able to get information through the network that is real information because it can't reach the energy level required to get there. Now, let me apply that to psychotic symptoms. So the guy's name, who I couldn't pronounce earlier, and colleagues looked at how attractor networks might have relevance to the positive symptoms of psychosis. One of the examples that they gave is that prolonged sensory deprivation can be reliably shown to produce hallucinations even in healthy individuals. But unhealthy individuals are more sensitive to it, so it can worsen delirium, for example. So their hypothesis is basically that a lack of sensory information causes the body to lower the activation energy that is needed to get to the higher energy state so that smaller signals can get through. So it can't hear anything, it can't see anything, and the brain wants to be able to experience the world. So it basically just lowers the threshold for getting sensory information. And that makes the curve look like this. So it's easier to get from the low basin to the high basin. Now this is important because you also change then the rate of your false positives versus your false negatives. And this is always a trade-off, right? If you want to get more information through, you're going to get false information through. But you're going to get more of the real information through at the same time. So some of the ways that this might work in, for example, like schizophrenia, is that there are demonstrable changes in GABA and glutamate signaling. So there's less NMDA signaling, and this is reducing the energy required basically to exit the active state, and it's reducing the overall excitatory nature of the network. But there's also less GABA signal, and this is reducing the energy required to enter that active state. So our normal states kind of look like this, but if you make those changes, well, you can get into a pathological state. So the way that they describe this is that a network with these characteristics will display loose and erratic jumps between low-frequency resting states and high-frequency active states. So it looks something like this. And delusions and hallucinations might share this kind of mechanism where there's instability in excitatory states resulting in false information. Another thing that might affect these states is dopamine. So we know that for people who have chronic psychotic processes, the levels of dopamine in different parts of their brain are either elevated compared to healthy controls or reduced. So in the parts of their brain where dopamine is elevated, it could change the signal to noise ratio in favor of the noise, in favor of false positives. And it does this by reducing GABAergic collateral inhibition. So basically, if you release more dopamine, it'll hit D2 receptors, and this will inhibit the GABAergic neurons, and the GABAergic neurons are inhibitors. So basically, it's allowing more signals to be sent because you need less energy to send them because the pyramidal neurons are not being inhibited by these GABAergic signals. You can have the opposite thing too. So in some parts of the brain, there is less GABA than you would expect in healthy controls. So what this might do is increase the energy required to enter the active state. And this potentially could result in some things like cognitive impairment, avolition, and even maybe affective flattening. So it's harder to get from your low energy basin to your high energy basin if you are deficient in dopamine. Now, this is a problem also in Parkinson's disease, we know, because dopamine production is reduced, and this results in movement disorders. And the way that it results in movement disorders is that having a deficiency of dopamine means that you're not inhibiting the GABAergic neurons in your corticostriatal thalamocortical tract. So you're allowing more signals to get through, and that is causing more signals to be sent down to your muscles, and that results in things like rigidity. So you're trying to move, there's too much signal, too many muscles are contracting, so it's very difficult to move. Or you get a tremor, like more signals are sent through than should be, and your muscles develop tremors. Now I want to move on to how attractor networks might work in affective disorders and with our basic motivation. So this is an AI-generated image that I got with a few prompts and then stuck them together in Adobe Photoshop. It's supposed to show a boy looking up at a bunch of apples in a tree. Now this is later in the fall, and all of the low-hanging fruit has been picked, and the boy has to decide whether or not to climb that tree and get that apple. Well, this might have a lot to do with things like if he had just had lunch, or maybe he's a migrant and he hasn't eaten in a couple days, and so that apple is looking really good to him. But how does he decide whether or not to take the risk of climbing that tree? So in our affective disorders, like depression, we know that our levels of motivation change. So when you're depressed, you have amotivation, or like an absence of goal-oriented behaviors, or a reduction in those, and a lack of energy. You may have anhedonia. That's basically an absence of pleasure-seeking, where pleasurable things just don't seem that motivating to you. You may even devalue our primary reinforcers like food or social connections. Mania, though, is associated with almost the opposite: like excessive energy, more goal-oriented behaviors, and hedonism, where you just look for pleasurable experiences. And excess value attribution, what that basically means is you think something has a lot more value than it does. It's a lot easier to get someone who's manic to, say, invest in some scam stock scheme than it is to get someone who is depressed. Because the person who is depressed just doesn't really care. But the person who's manic thinks that everything is great. Now, in both mania and depression, we have executive function problems. So there can be problems in our levels of attention, our ability to concentrate, our ability to reason. And sleep is impaired in both mania and depression. We think of mania as being a state where you don't sleep much, but in depression, you also often don't sleep much, especially in the classical varieties. So you go to sleep, you wake up early in the morning, you can't go back to sleep. With mania, you feel so much energy that you don't even need sleep often. Now, our limbic pathways have a lot to do with our motivation. They are our basic pathways. Limbic just means on the edge, and what this means is that all of these pathways that form our evolutionarily old system of do or don't do are still organized in a similar way. But in humans, there's a lot more going on there. And I took this image from physiopedia.com. So imagine that the boy sees an apple. What happens? Well, basically two things, at least. There's more than this, but I'm simplifying. You get information sent in a dorsal stream and in a ventral stream. The dorsal stream sends information up to the parietal lobe from your occipital lobe, where the basic visual information was processed. And in your parietal lobe, you determine things like the form of the object, its location, its size, its relative distance. But in the temporal lobe, you are thinking more about the identity of that object and its salient features. So what is important about that object? The ventral stream. So in the temporal lobe, where you're thinking about identity, you don't just think about identity, you send that information somewhere. And one of the places you send it is to the orbitofrontal cortex and also to the amygdala. Now, in the amygdala, this object gets associated with some sort of affective state, like, is this object something that scares me? Is this object something that generally makes me happy? Now, the amygdala can affect other brain regions, especially the hypothalamus, and this can initiate autonomic responses. So those can prepare us for action. It can get us excited, or it can make us afraid, but it can also make us freeze. But in the orbital frontal cortex, what we're doing is basically calculating the reward value of that object. Reward value can be thought of in many different ways. There's the primary reinforcement value. That's like, is this thing sweet or not? There's also the value associated with whether or not you're hungry, if it's food, for example. There's also value-based memories that are coming into play. Your current emotional state has a lot to do with how you value something that you see. Is this something that I need to deal with right now? And then there's top-down factors, and this is what things like advertising targets. Imagine you just saw a sign that said, "These are the most delicious apples in the world." How would that affect how your orbital frontal cortex values that apple? Now, this integrated reward value that has all that other information in it is then sent to the anterior cingulate gyrus. And then it's sent through pathways down to your brainstem nuclei. In your anterior cingulate cortex, you're processing reward value, and you're processing whether or not it was lacking in reward value, punishing, or whether or not it has some reward value. And there are different parts of your anterior cingulate that are specialized to receive those signals. But there's also something going on in that dorsal stream. So the boy has to locate that apple, right? It has to say, well, where is that apple? And is it even possible for me to get to it, for example? That information is processed first in the parietal lobe and then sent to your posterior cingulate. Before we were in the anterior cingulate, now we're in the posterior cingulate. The posterior cingulate acts kind of as a way station to and from the hippocampus, for example. This helps us with our spatial memories. It also communicates this spatial information forward to the mid-cingulate. So the ventral and dorsal streams align, and they align in that middle cingulate. So we were in the anterior cingulate, and it was connected to the orbital frontal cortex and the medial prefrontal cortex and the middle cingulate and the posterior cingulate and the amygdala and the insula and the autonomic brainstem nuclei. All this information is getting reduced down into a reward value that's final calculation happens in the anterior cingulate. And this is so we can facilitate emotion, motivation, and connect that reward value to our actions. Our actuator is actually the middle cingulate, so it is communicating with our premotor regions, basically, getting us ready to move or determining the risks of our behaviors. And this, in turn, is facilitating something called action outcome learning, which can be incorporated into our memory. Now, at some point, the apple's not going to have as much value. Say the boy got up the tree, he got a whole bucket full of them, he came down, he ate 4 or 5, and now he's looking at the apple and thinking something very different than he did in the beginning. He's either satisfied or maybe a little bit disgusted by the apple, but the reward signal stops at some point, and our action outcome learning updates. So it's no longer saying, do something about this apple. It's like, do nothing or even avoid the apples for now. Now, I'm going to try to incorporate all of this into ideas about depression and also obsessive compulsive disorder, but I'm going to start with depression. Depression has been described many different ways. Freud described it as anger or aggression turned inward. Jung conceptualized depression as the depleted ego's experience of itself. Beck argued that depression has something to do with dysfunctional or distorted beliefs that are incorporated into self-defeating schema. Mahler interpreted depression as object lost in fantasy, that is to say, intrapsychic conflict. So you haven't actually lost anything, but you just feel like you have. People who are depressed lack motivation. They lack energy. They lack basic drives sometimes. They ruminate. They feel hopeless. They feel guilty. The difficulty with concepts like Freud's, Jung's, Beck's, and Mahler's is that really they're mostly metaphorical and often untestable, or mostly untestable. They really offer little help in elucidating the actual neuropsychiatric mechanisms. So Edmund Rules at the Oxford Center for Computational Neuroscience has tried to attach mechanisms to depression using attractor states. So he proposed that depression is a non-reward punishment attractor state. It's initiated by our lateral orbital frontal cortex, and it spreads through the brain to our top-down executive networks, and those exert a negative bias on our cognitions, and that further biases the orbital frontal cortex and helps to bias our reward centers toward non-reward states. So basically, increases in the lateral orbital frontal cortex combined with decreases in the medial orbital frontal cortex bias our affective system, and this prevents reward-seeking behaviors. And he said that this is similar to what happens in learned helplessness. So imagine you see the thing on the left and the thing on the right. If you're looking at the ice cream on the left, you may be thinking, yum. If you're looking at the emoji on the right, you may be thinking, I'm going to avoid that thing. So your lateral orbital frontal cortex calculates signals that having to do with non-reward and punishment expectations. So when something is determined to not be rewarding or even punishing, the values that are placed on that, the extent to which we think it's punishing, happens in the lateral orbital frontal cortex. It's the medial orbital frontal cortex that calculate signals that have actual reward value. So you could say if it's positively rewarding, the medial orbital frontal cortex sends those signals. If it's negatively rewarding or punishing, the lateral orbital frontal cortex is sending those signals. So in Roll's model, the lateral orbital frontal cortex is biased toward false positive non-reward signals. So it's sending a lot of signals that are non-rewarding. And this could be a predisposition because of genetics, maybe chronic stress, adverse experiences in your past, but also like brain lesions, like cancers. But it can also be executive biases like devaluation, things people have said to you that you've incorporated into your thoughts. And it can also have something to do with the lack of activity in the medial orbital frontal cortex, because the medial orbital frontal cortex is supposed to inhibit the lateral orbital frontal cortex and vice versa. Overall, what this does is lower the activation energy so that false positive signals, non-reward signals, can get through. And this results in things like rumination, guilt, and can even result in thoughts of being worthless. Now, the medial orbital frontal cortex is supposed to be sending reward signals, but it's not. So it's biased toward false negative reward signals. Even when it sees something rewarding, it's not telling the anterior cingulate that. And this also can be the result of things like genetics or chronic stress, etc. But in the end, more energy is needed to send the reward signal, and less energy is needed to return to the low energy basin. So it's just hanging out in a low energy basin, having difficulty sending reward signals, and when it does, it more easily loses that signal. We know that the orbitofrontal cortex is not sending normal reward signals, and signals are not only being sent to our anterior cingulate, they're also being sent to our nucleus accumbens. So when we gain some reward, we're supposed to be sending signals to the nucleus accumbens that tells it that. And that's supposed to feed to other brain systems, and especially this area of the thalamus called the lateral habenula. If it doesn't send signals to the lateral habenula or inhibit the lateral habenula, then the lateral habenula is going to think that something not good happened, and it's going to send signals to our rostromedial tegmental area, which is then going to inhibit our brainstem nuclei. That's a long way of saying that if non-reward signals go through, our brain tells itself to kind of shut down, to become less motivated, or to withdraw. So how this affects depression is kind of obvious. You are not getting reward signals, you become unmotivated, you start to think that you're just worthless, and at times even become potentially suicidal. But how would this system affect obsessive compulsive disorder? In this disorder, we think that the orbitofrontal cortex is basically sending aberrant repeated signals to the cingulate cortex. So instead of it like not sending signals or being biased toward non-reward signals, it's just overall sending aberrant signals, signals that it shouldn't be sending to the anterior cingulate cortex. And because it's sending continuous signals, those signals might be to like gain a reward. So if I do this, something good is going to happen. Or it might be to avoid a punishment. Like if I do this, something bad won't happen. I have this feeling that something bad is going to happen. I have to keep doing this over and over and over again. And we know that these signals get sent to the mid-cingulate and premotor cortex is activated, trying to figure out what to do about it. So this sounds a lot like OCD. You get a lot of signals coming from your orbitofrontal cortex to your anterior cingulate. These signals are aberrant. They're saying things like you need to do something or something bad is going to happen. And so you plan out doing it and then you do it. And because the signals don't get suppressed, you never satiate basically your orbitofrontal cortex. You just keep doing the same thing over and over and over again. And this can affect also how you feel. If you're getting false positive gain of reward signals, like you do something and then you're like, okay, I've done that, something bad's not going to happen, that could result in a false positive reward signal, which is going to make you feel better, basically. But if you're getting false negative gain of reward signals, like you're doing something and doing it again and doing it again, but you're not getting that reward signal sent through, then you could become despondent, less motivated, sad. So I want to talk about these networks in relationship to depression and OCD treatments. So we know that brain connectivity changes with depression treatment. And there was a 2023 review that reported this figure, and I'm showing here that there were some predicted changes in certain brain regions given like pharmacotherapy or psychotherapy or ECT or TMS. The blue dots here, like these dots here, were changes that happened. The purple dots were changes that were both predicted and happened. In general, they found, among other things, changes in functional connectivity of the orbital frontal cortex very frequently, but also the dorsolateral prefrontal cortex, which is a main hub of our executive network. Psychotherapy really did not have robust evidence for change, so all this predicted change, but not measured change. I should mention here that it was far less studied as well, so there may be changes happening, we just don't see them because we're not looking for them. So how would ECT work? Well, it could potentially reset our attractor states. Successful ECT has been associated with reduced activation of the orbital frontal cortex. But it may also reduce those negative reinforcing signals. By negative, I don't mean like lack of something, I mean like this is a bad thing. You are a bad person. And it may also just reset our affective networks. TMS may work in a similar way if you excite the dorsolateral prefrontal cortex with transcranial magnetic stimulation. This has indirect effects on the orbital frontal cortex and also on the nucleus accumbens. So this can result in basically an normalizing reward-seeking behaviors, normalizing our affective states. It's also been shown that like TMS of the orbital frontal cortex is a possible treatment for OCD, but it's not widely used that I know of right now. Now, when someone is very suicidal or even has treatment-resistant depression, one of the things that we can use is ketamine, and it seems to work really fast to change someone's mood. So it's proposed that ketamine's efficacy may be related to its ability to just quash attractor networks temporarily. So what that means is that because it is blocking excitatory transmission, it's increasing the signal needed in order to get from your low basin to your high basin. So you're no longer sending those aberrant non-reward signals as easily. It basically kind of resets the network for a while. But we also know that ketamine tends to wear off in most people after a few days, and they just sink back down into the same state that they were in before. Now, SSRIs and SNRIs work a little bit differently. So instead of going to a state and then dropping back down, we use these medications chronically, and it slowly changes that state. And we think they do that by reducing the excitability of the pyramidal neurons, and they do that potentially by increasing GABA transmission in the cortex. And all of this, in turn, kind of counters the lateral habenulous suppression of like serotonin and dopamine release in the brainstem nuclei. So we get less suppression of our brainstem nuclei and more control over our orbital frontal cortex. So looking at this in that network perspective that I put up before, electromagnetic and pharmacotherapies tend to work either like on the extracellular or cellular levels, basically resetting these small cellular networks. They also seem to have obvious effects on whole brain functioning. And this, of course, affects our behaviors, and then our behaviors affects how we exist in society. Psychotherapies kind of come in here. They target our behaviors and our thoughts. So we either try to reduce a behavior or we try to change the way we think. And this is potentially affecting our autonomic responses and other things in the brain, and that's changing potentially our attractor states. We can also reduce social distress, or say reduce poverty, or reduce hunger. And this is going to change how people think and how they behave, and this is going to change how they feel. And so in general, we have a population that will have healthier attractor states because they're not getting a lot of negative bias from their executive centers. So basically, I want to point out that the things we know determine what we believe in the questions that we ask. And Bayesian reasoning is really the only thing that we have that deals optimally with uncertainty. And that's why our neurons work the way they do. They have a setting, but they're always a little uncertain. They can change that setting back and forth. And sometimes changes in those settings result in neuropathologies. We can't really develop new treatments until we have new concepts. And these network models are some of the newer concepts that we have. The way that we've historically conceptualized psychopathology, I think, has really reached its limits of effectiveness. So the clinical utility of most of the medications that we use for targeting like certain receptors, I think it's reached kind of an asymptote. We're getting diminishing returns now from simply making new medicines. Psychotherapies are targeting top-down effects, but they really have limited power to affect the lower regions, and they have a very limited power to teach us about the neuropsychiatric mechanisms of disease. So that's why, in the end, I feel like we need to adopt a more Bayesian perspective. We need to think about the brain in terms of being basically a Bayesian calculator. And I think this is going to help us in the future to change what we know about disease and to treat it better. Thank you for joining me. I am Dr. O'Leary, and this has been an episode of Psydactic. Welcome to Psydactic, Residency Edition. It's not Residency Edition anymore.